Okay, guys. So, hello, welcome to the histology study along. I am going to start with the first lecture. It's going to be about the cytoplasm, and I'm going to study along with you. I'm going to read the book Junquera Histology, and the first chapter was quite basic of specimen preparation and microscopy for the histology. I would like to go from chapter second, the cytoplasm. We will study about cell differentiation here. We'll study about the how the plasma membrane works. We'll see about the cytoplasmic organelles in the cell. We'll also study the cytoskeleton. Let's get along. I hope you can see the screen and everything, and I hope my voice is audible. Okay, so cells and extracellular matrix together comprise the tissues that make up animal organs. Okay. In all tissues, cells are the basic structural and functional unit, the smallest living part of the body. So basically, cells together make a tissue, tissue together make an organ, organ together make an organ system, and different organ system work together for a functional human. So the functional body. So this and it all boils down to a functional unit, the cell. Animal cells are enclosed by cell membrane and are eukaryotes, each with a distinct cell membrane enclosed nucleus and surrounded by cytoplasm. So cell has a cell membrane, and in a cell there is a membrane enclosed nucleus, then rest part is called cytoplasm. Fluid containing a cystine of membranous organelles, non-membranous molecular assembles, and cytoskeleton. What does cytoplasm consist? Some are membrane organelles, some are non-membrane organelles, and we have cytoskeleton. In contrast, the smaller prokaryote bacteria typically have a cell wall and lack nuclei and membranous cytoplasm. So we don't need to care about prokaryotes. Let's check what is cell differentiation. So the average adult human consists of nearly 40 trillion cells according to the best available estimate. Okay, so these cells exist as hundreds of histologically distinct cell types, all derived from the zygote and the single cell formed by the merger of a spermatozoon with an oocyte at fertilization. So this takes us back to a little bit of embryology, that how the first cell was derived from the first ever fertilization of a sperm and an ovum. The first zygote cellular division produced are called blastomeres, and as part of the early embryo's inner cell mass, blastomeres give rise to all tissue types of the fetus. Explained to the explanted to tissue culture, cells of the inner cell mass are called embryonic stem cells. Most cells of the fetus undergo. Okay, so the after the first division, the cells that are formed are called blastomeres. We have a sperm, we have an ovum, they have fertilization, and a zygote is formed. And the first differentiation that it undergoes in the cells that are formed are called blastomeres, and they later develop all the tissues for the fetus. And explanted to tissue culture, cells of the inner cell mass are called embryonic stem cells. We will study what are embryonic stem cells in the later part, and embryology has we also will take a look at embryology and on this channel, and we'll study much in detail how the blastomere cells are arranged, and which of those the inner parts are called the embryonic stem cells, etc., and how they differentiate later. Most cells of the fetus undergo specialization process called differentiation, in which these predominantly express sets of genes that mediate specific cytoplasmic activities, becoming efficiently organized in tissues with specialized functions and usually changing their shape accordingly. So we'll take a look of what is differentiation. We'll just highlight this part so that we know for it later, that what does differentiation mean. So some cells of the fetus undergo a specialized process called differentiation. What is this specialized process? So in which they predominantly express sets of genes that mediate specific cytoplasmic activities, becoming efficiently organized in tissues with specialized functions and usually changing their shape accordingly. So the initial changes, the differentiation, is predominantly expressing some specific set of genes. This why this only this specific set of genes, because they are important for the early growth of the earlier cells, the cytoplasmic activity, and start to the initial tissues that later on develop a complete organism. For example, we have an example here as well. So for example, muscle cell precursors elongate into long fiber like cells containing large arrays of actin and myosin. All animals, so the precursor cells are basically produced here, and each division gives out a different type of cells which will later give rise to a different type of tissue in our body. All animal cells contain actin filaments and myosins, but muscle cells are specialized for using their proteins to convert chemical energy into foreseeable contractions. We know the role of muscles, actin and myosin. We'll study about muscles in detail in histology. Major cellular functions performed by specialized cells in the body are listed in table 2.1. We will also check table 2.1. Okay, so here is a general activity that a human does and what muscles and specialized cells lead those activities. We have this table here, and that's why they are differentiated for their specific role. Major cell functions predominantly Major cell functions performed by specialized cells in the body are listed in table 2.1. Okay. It is important to understand that the functions listed there can be performed by most cells of the body. Specialized cells have greatly expanded their capacity for one or more of these functions during differentiation. Changes in cells microenvironments under normal and pathologic conditions can cause the same cell type to have variable features and activities. Cells that appear similar structurally often have different families of receptors for signaling molecules such as hormones and extracellular matrix components, causing them to behave differently. So let's see. So what we saw in table 2.1 is not the complete truth. Like we know that muscles carry out the movement, but can only the muscle cells carry out the movement? No, each cell can do pretty much all the activity, but each cell has a specialized role, and that is the basis of differentiation. Also, we read that cells appear differently because they have different family of receptors in their cytoplasm, and they react differently to different stimuli. And here is an example. Because of the diverse arrays of receptors, breast fibroblasts and uterine muscle cells are exceptionally sensitive to female sex hormones, while most other fibroblasts and smooth muscle cells are insensitive. Please, is my sound not audible? Hold on, guys. Give me a second.